Thanks a lot for inviting me because it's an honor to be here. And also learn from you as neuroscientists because I'm actually dietary, more experienced in dietary studies, but do not have that much expertise on mental health. So if we could learn from each other, that would be great. Nicole de Wit, I'm working at Wageningen Food and Bio-Based Research, which is part of Wageningen University. And we're mainly doing a lot of dietary intervention studies to study the interaction between food and human health. And there's a special focus nowadays on mental health. This is really hot topic. And we are doing a lot of work with companies, together with companies, especially food companies. So today I want to tell you a little bit more on the capability of food to modulate the gut-brain axis. So it's a little bit different angle than Peter Joris introduced. I'm mainly going to focus on dietary fiber and the effect of dietary fiber and some other related compounds and the effect on the gut-brain axis. And actually, dietary fiber drives this influence on mood, let's say, via your gut microbiome, at least partly via the gut microbiome. So that's the bacteria that are in your large intestine and producing all kind of metabolites when they ferment the dietary fibers in your diet.
I need to interrupt a brief second. Yeah. I want to double check that it's recording because I couldn't see it from there. Just 1 second. Yeah, perfect. Okay. Yeah, sorry for the interruption.
No problem. So I'm mainly going to focus on dietary fiber and also the prebiotics. And prebiotics are the dietary fibers that are fermented by bacteria in your large intestine. So it's not the complete dietary fiber population, but it's just a part of it. I will discuss this later. And I will focus on the fermented foods and the pro and postbiotics, which are more about the bacteria in our food. First of all, why are we studying the dietary fibers? Well, in general, there's a big dietary fiber gap, not only in the Netherlands, but I guess worldwide, because we are far from the recommended intake, as you see here. So there's still quite a gap to overcome, and we know that dietary fiber is really healthy to consume. It has all kind of health effects, like it controls your blood sugar. It is an impact on satiety. So maybe the mixed nuts intervention from that was just discussed is also partly elaborated by the dietary fiber, the effect on satiety. But what are the effects on mental health? Well, in general, you have two different kind of effects of dietary fiber. It's microbiome independent. So there are certain characteristics of dietary fiber that they can influence the volume of your stool, let's say, and they can bind all kind of compounds like, for instance, cholesterol or bile acids. And in that way, they can affect your health and they can affect your mental health. But it's also mainly these effects are driven by microbiome dependent effects. So the bacteria in your large intestine are involved in it, shown here. The dietary fibers are fermented, so broken down in the large intestine, and they result in short chain fatty acids that can be taken up by the body, and they are kind of energy source for your large intestine, but they can also signal to other parts of the body, like for instance, the brain. So first of all, something more about dietary fiber and prebiotics. The ASAP, the International Scientific Association of Pre and Probiotics, have a strict definition for a prebiotic, saying that it's a substrate that is selectively utilized by host microorganisms and it confers a health benefit. So both are important. They are present in our food, especially in fruit and vegetables. This is just an example. These dietary compounds of these food products are high in prebiotic fibers, but of course they're also high in all kinds of supplements you can find in the drugstore nowadays. In general, in research, we mainly looked at the effect of prebiotics on the gut microbiota composition. So how do they affect the number of specific bacterial strains in our large intestine? And that's actually the old way of looking at it. So looking at compositional changes, if you have an intervention with a specific dietary fiber, and what would be the effect on health? But as a lot of these bacterial species have a kind of overlapping functioning, just looking at composition does not give us the solution of this puzzle, let's say. So nowadays we're looking much more on the capacity and the activity of the microbiome. So we're looking at genetic profiles of your total gut microbiome, seeing which kind of enzymes are in there, for instance, and that can help us degrade all these kind of fibers, and which metabolites are produced. So which short chain fatty acids are produced, and all kind of other health beneficial metabolites. And in that way, we are looking at how does this link to health. And then we are talking more about the structure-function relationship. So how does this work? This is just an example. There are a lot of genes in this bacterial population. They're called the carbohydrate active enzymes, and they help degrade all kind of different types of dietary fiber. And these K-zymes are their genetic profile is a kind of like a fingerprint. So every individual has a different K-zyme profile. So if you know what is your specific K-zyme profile, you can maybe think about what dietary fiber is most beneficial for you, because you have the enzymes to degrade these fibers in your large intestine. So in that way, you can predict which kind of beneficial metabolites are produced and how this will affect your health. But we are far from the solution from this. So we know there are K-zymes in your bacterial profile, let's say, but how they link to specific dietary fibers is not known yet. So we are at the start of this. So at Wageningen Food and Bio-Based Research, we have currently running a new project. It's called FiberMe. And with in silico and in vitro studies, we try to study the connection between specific dietary fibers and these K-zyme profiles in your microbiome. And in this way, we want to give you a more accurate dietary advice on which fibers are more beneficial for you than others. This project just started, so hopefully within 4 years, we have more information on this. Then a little bit more on fermented foods and pro and postbiotics. The ASAP definition is that especially probiotics are live microorganisms, and again, they confer a health benefit. So where can we find it in our dietary pattern? These are the food products where you can find live bacteria in there, like for instance, yogurt, kefir, kimchi, and much more, kombucha. I guess the natto, I don't know if you ever had it, but I think it's more consumed in Asia. In general, the European people do not like it that much, I think. And of course, again, there are a lot of supplements on the market with these probiotic strains. And highly linked to this are the postbiotics. This is actually a new category linked to these biotic supplements or these biotic foods. And it's a preparation of an inanimate, so not live, microorganism and their compounds. And again, they confer a health benefit. Similar food products, because when live bacteria are in these food products, also the metabolites are still produced. But next to these metabolites, you can also think about the microbes themselves that are not alive anymore because, for instance, because of pasteurization of yogurt or these kind of things. But also the enzymes that are in these bacteria that are coming out, and the short chain fatty acid and all kind of other metabolites. First of all, an example now on a study that we performed at Wageningen Food and Bio-Based Research. This was an intervention study in patients with irritable bowel syndrome. And in general, these patients have a lot of stool complaints. And in this study, we especially focused on patients with constipation-related complaints. We had 60 people per arm, and we gave them first 4 weeks of no intervention, just to have like a baseline ID on the complaints of these people. And then the last 4 weeks, they had a dietary intervention with a prebiotic and a postbiotic. The prebiotic was an acacia fiber provided by Nexira, a company in France. And the probiotic was a bifidolactis bacterium provided by VCare, a company in China. And we had a placebo control. Really a good question, because I think in these kind of studies, we really need a good placebo control. And this is really difficult. We measured all kind of symptoms like the POC-SIM. We measured quality of life. We measured symptom severity of IBS complaints, and also food frequency questionnaires, so their habitual dietary intake, and if we do see changes in that. Next to that, we also had daily questionnaires via an app, and we, for instance, requested on stool frequency and stool consistency and GI complaints, which is also really important in this population. What we found is that the pre as well as the postbiotics had a really big effect on stool frequency. So the increased stool frequency for the prebiotic, this was even significantly clinically significantly different. This is FDA guidelines saying that if you have an increase of more than 1 stool per week, it's clinically significant. So patients really experience a relief from this intervention. But also the probiotic had a significant improvement in their stool pattern. And of course, we also looked at a more mental aspect, like severity of complaints, but also quality of life. And in general, you see that in the first 4 weeks, the observation period, there was hardly any effect. But in the last 4 weeks during the intervention, we saw significant effects, and especially for the probiotic intervention on symptom severity. But what you can also see in these graphs is that there is a substantial placebo effect. So you really have to have an accurate study design and take along this placebo control group to really say anything about the treatment effects. Then something on the probiotics, or actually the postbiotics. So this is a really nice example of how a compound that started as a probiotic supplement is now converted to a postbiotic supplement, because they found that not only the live bacteria of this Akkermansia muciniphila has a health effect, but also if you pasteurize this bacterium, so when it's not alive anymore, it has similar health effects. And even if you're only looking at the extracellular vesicles, so you do not have the complete bacterium anymore, you still have the same health effects. So you can imagine for practical applications, it's much easier to introduce a pasteurized bacterium in your food products, or only the extracellular vesicles, than really the live bacteria. So in this case, a probiotic supplement changed into a postbiotic supplement with similar health effects. Then another example of a study that we performed quite recently at Wageningen University and Research Center together with VU Amsterdam. It was a kind of citizen science approach. So really not controlled. People were their own scientists, let's say, because we did not tell them exactly what to eat, but they got recipe booklets to increase their fiber intake or to increase their fermented food intake. So we had 3 intervention arms with approximately 50 people per arm. The first 2 weeks, they only get the recipe booklets to increase their fiber intake or fermented food intake, or they got no intervention, just refer to the website of the Dutch Nutritional Center to say, look at this adapt a healthy diet and see what it can do for your microbiome. So actually, that's no advice. People think it's really difficult to work with that. And after these 2 weeks of dietary advice with the recipe booklets or the reference to the website, they got an additional food supplement. In the fermented food group, this was a kombucha beverage from Anybiome. In the high dietary fiber group, this was whole fiber, a kind of dried chicory root with prebiotic potential. And in the control group, they got maltodextrin as a placebo. In the end, and at start, people did not have that high fiber intake, so beneath 20 grams a day. And with the dietary intervention, we reached 27 to 37 grams a day. So this is a significant improvement. And for the fermented food group, they started with lower than 3 portions per day of fermented food, like the yogurt or the kimchi or the kefir. And in the end, they had 6 to 9 portions per day. Okay, we did at week 0, 2, and 8. The participants collected fecal samples. They sent it in to a company that did the sequencing. They got back their personal report. And of course, we got back the raw data so that we could work with that as well. But we really want to do this as a kind of citizen science so people can get feedback on their own changes on their microbiome. They also did blood sampling with dried blood spots. So just a finger prick, dried blood spots. They sent it in to us. And we also looked at changes in immune function. Okay. First, we looked at the effect of the gut microbiome. In purple here, you see the changes from week 0 to week 8 for the fermented food group. And in gray, you see the changes in the placebo group. And when the purple line exceeds the gray line, then that's an increase of this bacterial strain. And if the purple line is lower, more to the center of than the gray line, then it's a reduction in this bacterial strain. So you see that there are some significant differences between the placebo and the high fermented food group, but they are not that pronounced. But maybe that is not so unlogical because a postbiotic also exists of a lot of metabolites, and they are probably not even reaching the large intestine. They are already absorbed in the small intestine. So maybe the large intestine and just fecal samples are not the right sample to look at. And in future study, we are now thinking about implementing these sampling pills. This is quite innovative technology. So you take in these sampling pills. It's just like a capsule with medicine. And they will sample in your small intestine. You poop it out, you collect them, and then we can extract the sample and look at, for instance, microbial changes, because also in your small intestine, you have already some bacterial species there, not as much as in the large intestine, but it's still there. And you can also look at all kinds of metabolites. So we did not find, oh, oh, interesting. There are completely different data in here. Okay, but just to let you know that we did not see that much effect on the gut microbiome, let's say, but we did see an effect on immune modulation, especially T-cell related responses are really induced. So we do not know exactly what this means for health reasons, because you see an increase in these T-cell responses. Maybe the body is more ready for all kind of invasion of bacteria or infection, but at least you can see that especially the high fermented fiber is capable of modulating the immune function, and this is not seen in the high dietary fiber group. Actually, here on the right, there was a similar spider web for the high dietary fiber group, but it's really not representative anymore, so I can just tell you what happens. On a dietary fiber background, you see that the changes in microbiota are much bigger compared to placebo than with the high fermented food group. And here you really saw that, yeah, this is also really small. But what we found is that bacterial strains are increased that are really linked to this prebiotic fiber that we gave in the study product. Previous study already showed which bacterial strains were influenced by this prebiotic, and these bacteria are part of a kind of trophic chain, so they are needed to degrade this specific fiber and convert it into butyrate. And butyrate is a short chain fatty acid that with high beneficial health effects. So on top of this dietary high fiber dietary background that we have with the recipe booklets, you still see a similar effect of the food product. So sometimes companies are really worried that you have a high fiber background already and then the supplement will not do that much. But we show that even on a high fiber background, your prebiotic compound can still have a large effect on, for instance, your gut microbiota profiles. And another thing that we found with this high dietary fiber intervention is that you see an improvement in sleep quality. So this brings us more to mental health and mental well-being. So sleep quality was already improved after 2 weeks of intervention, but it got significant after 8 weeks of intervention. And we also had a follow-up 3 months later. So after 8 weeks, the dietary intervention stopped. We had an additional measurement at 3 months later, and you still see an improvement in sleep. So it is a sustained effect of this high dietary fiber intervention. Of course, we do not know, unfortunately, we do not know yet what is the exact mechanism behind it. And what was discussed by Peter Joris as well, it's really hard to do mechanistic research in the brain. So it's always correlative measurements. But we think that especially these short chain fatty acids, so that are the metabolites that are derived from fiber fermentation, exert this effect in the brain. They can, via the blood flow, go to your brain and they have an effect on your sleep quality. But as said, human intervention studies are largely lacking. A lot of these studies are focused on animal studies because you there can better measure mechanistics. Human studies are largely lacking so far. So this brings us more to the gut-brain axis. You have different bidirectional routes how the gut and the brain can communicate with each other. Like for instance, via the vagus nerve. So with neurotransmitters that give an effect on the nerve system, but also via the circulation. And then you can think about all kind of hormones, like stress hormones, like cortisol, but also the gut hormones seem to signal back to the brain, as we also discussed with GLP-1, for instance. But all kind of cytokines are also known to signal from the gut to the brain, and maybe also the other way around. So most of the studies that we have so far on mental health are linked to depression and anxiety and are mainly performed in animals. And the human studies are still limited. And if we see human studies already, there are a lot of variable results. So some of these studies report reduced symptoms and others report no effects. On the other hand, you do not see that much of studies that report detrimental effects. So or you see no effect or it's an improvement in mental health. So that's promising. And also really important, as said before, the placebo effect. We also saw it in our study because if you look at the alpha diversity, so how diverse your microbiome is, in general, the message is the more diverse your gut microbiome, the healthier it is. But we also saw that a placebo had this effect. So you also see an increase in diversity. This is kind of strange. So maybe the brain already signals back to the gut, hey, you're involved in a human intervention study. There should be a beneficial effect. And this already has an effect on your physical objective outcome, gut microbiota diversity. So interesting, but a lot of work to do there, I think. Why are these human intervention studies largely lacking? well, it's quite difficult to have a good study design. For instance, the sample size is often too small. Yeah, there are a lot of reasons behind it. Sometimes it's project budget. I think everybody understands this. Sometimes they had the wrong primary outcome where they did the power calculation on, but sample size is really important. Varying inclusion and exclusion criteria. So you have various degrees of depression, for instance, or what kind of mental health stage you want to include. Do you also want to include inclusion and exclusion criteria for your dietary status? So there are a lot of variations in these inclusion and exclusion criteria. The duration of the treatment is always different. So for Peter Joris' story, they had a six-week intervention with the mixed nuts, but sometimes a longer intervention does not per se mean that you have a bigger effect, because sometimes a shorter intervention, people are still aware and are really compliant to the dietary advices, and then the 4-week is, for instance, a better effect than the 16 weeks. Also take that into account. And then your primary outcome. Most of the time, mental health is assessed by questionnaires, but you have a lot of different questionnaires. So that also gives a lot of different outcomes. So it's hard to draw solid conclusions on the effect of food on your mental health status. And a lot of studies do also not have a follow-up effect. So after you stop your intervention, what happens with the effect? Is it sustained, or will it only drop down after you have the dietary intervention? So all kinds of things to take into account if you're performing a human intervention study. Nowadays, we have two studies running on food, the intervention between food and mental health. That's the Oh My Gut project. That's mainly focusing on the effect of food on stress. It's a 3-year study, and we just started last year. We're now submitting a medical ethical protocol, and if it's approved, we hope to start the end of this year in 240 people with 6 intervention arms. And the Food for Mood project, it was just granted. This is mainly focused on students. Students also have all kinds of mental issues, and we hope with a more plant-based diet, with on top of that supplements that boost your mental health. We hope to study this and see some effects of food on mental issues in students. And then finally, some acknowledgements. Colleagues from Wageningen, but also from VU Amsterdam, and of course all the companies that are involved in these projects that I presented today. And finally, I want to draw your attention to this a new initiative. It's called Buikbelang, or in English, Gut for You. And it's a collaboration between 3 partners. It's the Dutch Digestive Health Fund, together with UMC Groningen and Wageningen University. And it's really all about understanding the gut microbiome and its link with human health. And it also tries to convert all kind of knowledge that is provided or acquired by the universities to make the translation into the clinic, to inform healthcare professionals on how they can implement food, postbiotics, these kind of things in the clinic and help their patients with it. So if you're interested, there's also a podcast. Unfortunately, it's in Dutch, but via the QR code, you can directly link to these podcasts and learn a little bit more on this Buikbelang initiative. And that's it. Thank you.
Thank you very much. Super interesting and stimulating talk.
Yeah.
Some time for questions. I have one that is a little bit tricky.
Yeah.
So obviously there is quite some push also from industry on the development of pre, pro, post, and symbiotics and so on, right?
Yeah.
But if you had to express your unbiased opinion, then comparing, sort of diets rich in fibers as compared to the use of supplements, also utilizing some knowledge from the scientific literature, then what would you think and what would you sort of recommend?
Yeah, well, really good question. I think in general, as a scientist, I would say first try to make the dietary changes from the whole diet perspective, but we see that a lot of people do not reach the recommendation of dietary fiber. So if you can help them with providing supplements, let's say, and these are just also dietary fibers, it's nothing strange about it, then that could help people to reach the recommendation and improve their health. But in general, first try to do this via your diet. And we also saw that with the recipe booklets, if you really guide people how they can do it, because a lot of people want to do it, but they do not know where to start. And if you can help them with making small changes, for instance, going from refined pasta to whole grain pasta, these small steps, this already helps in increasing their fiber intake. As for the probiotics, this is actually a kind of different field. I feel, to be honest, I think if you're healthy, you do not really need the probiotics, because dietary fiber is even more effective in modulating your gut microbiome and helping you to get healthy. But for certain patient groups, it can really help. It can really relieve their complaints. So in that way, in healthcare situations, I think the probiotics can really be an additional, not cure, but it can support health benefits.
Yeah. Any questions? Yes.
I got just a comment on this. My impression is generally that relying on supplements or probiotics instead of a good diet may also make people thinking that they can eat whatever they want, because at the end they will eat the supplement and they will eat that, which I don't think is the correct way to use.
Yeah, totally agree.
So maybe the dietary indication is more healthy also because you avoid eating other things while using supplement or probiotics is a kind of way to say I can eat whatever because at the end I will fix it.
Actually, yeah. Part of it is true, I guess, but as Peter Joris also said, with food you can probably not cure all kind of diseases. You can just mostly prevent all kind of diseases. And I totally agree that with dietary changes, it's better to do it with the whole diet and make changes there than just with the supplements. But sometimes you can support people in living a little bit more healthy. Yeah. It can never overrule. And all these kind of supplements like vitamins and all kind of other, I'm a little bit skeptic as well. If you're following the Wheel of Five, I think you're having a healthy diet and it will prevent already a lot of diseases. But sometimes it can just be the additional push for people to get them a little bit more healthy. Yeah. And for patient groups, it might be a different story. And that's why we are also thinking about how to improve the dietary advice for people with these K-sign profiles. If you can better advise people, this additional dietary fiber or prebiotic is really helping you for your health, that's better than just giving any prebiotic supplement, because in the end we do not even know how effective it is, because it might be different for you than for your neighbor, let's say. So in general, I totally agree. So supplements should never be an excuse of not doing anything about your dietary pattern, but it can help people to be a little bit more healthy. Yeah.
So maybe as a follow-up, like how far do you think we are to this approach that is more like precision medicine, right? That is knowing more or less your metabolism, the diversity of your gut microbiome. Okay, then I can target specific strains in the supplements that I sort of advise. But, I mean, how realistic it is given what we know now?
Yeah, I think the personalized approach, we are far from it, because every time we think this is the solution, then new questions pop up. So I do not think we have the solution within the first 3 or 4 years, let's say. But for targeted nutrition, it's different. For specific target groups, like for instance, this IBS population, if you're looking at gut microbiome composition, you see that they're often lacking a lot of bifidose. They are have a reduced number of bifidose in their gut microbiome profile. If you then give a probiotic specifically with bifidobacterium, this really might help them to relieve their symptoms. So targeted, we are already a little bit on the way. Personalized, I think we still have a lot of work to do. Yeah.
There was a question there.
Yeah. I was about to ask, for example, for different groups, I suppose the fibers necessity is different, like the requirements. We all lack of that, but also they should be different between ages or back in this case as well. Like maybe women need more or men need more. And the value that we are very behind of what we need. Actually, which value was compared, or which is the reference?
Yeah, that's a good question. I think every country has its own recommendations, but largely it's about 35 grams of fiber per day. In general, they say it's like 14 grams per 1,000 kilocalories. And then women need less than men because women need less calories than men. So in general, if you recalculate, in the Netherlands, we say women need 30 grams of fiber per day and men need 40 grams of fiber per day. I think in general, we are at 21 grams a day. So we need to double our intake. This is the gap that we need to overcome in the Netherlands. But we did already improve in the last decade, fortunately, but it's just small steps.
Yeah. Yes. Oh, sorry.
I was just wondering, how fixed are the KSAP profiles? Because you mentioned that according to the profile that you determine what you should eat more of, but how much can you actually manipulate your profile based on what you eat or even other factors?
Well, at least this research now is not so much focused on how can you manipulate this casein profile. It's just more that you can track your casein profile, so you can assess it based on your stool sample, and then you can make a dietary advice based on it. So this is your casein profile, and then this type of fiber is best for you because your casein profile is fit to degrade this kind of fiber. I think, and maybe you can still modulate it with specific fibers, but if you do not have a bacterial strain in your gut microbiome profile at all, you can feed the dietary fiber as well, but it will never never grow. It's not there. So I think the main thing that we're working on now is assessing the individual casein profile and then link this to a personalized dietary advice.
Yeah, I was wondering if you have any studies in patients like with obesity, because I was thinking in that clinical population, behavioral change is particularly difficult, especially in terms of healthy eating, healthy diet. So maybe it can support, not cure, as we were saying before, but maybe promoting a healthier lifestyle, maybe gradually help them change their diets.
Yeah. No, definitely. And I think with obesity, it's also seen that they have a kind of different profile on their bacteria than more leaner persons, let's say. On the other hand, it's not linked to specific bacteria, and we do not know on specific casein profiles, for instance. I think obesity was one of the first diseases that was linked to gut microbiome. But it's not that easy because obesity has a lot of different causal factors and changes in dietary lifestyle or dietary habits, let's say, and lifestyle. It's really difficult for people. And what we did is that we also developed a kind of web portal, and then we asked people their habitual eating pattern. And then with this web portal, they can make the switches to more fiber-rich foods. And in the end, this worked. Also, 3 months later, they still implemented more fiber-rich foods in their diet. But you need small steps, because if you get a top-down advice, completely change your diet, this is really, really difficult.
Yeah.
But it's an interesting and relevant target population because there's a lot to gain. Yeah.
Maybe I ask the last one. I mean, partially clarification question. So one of the results was also that there was no effect on the gut microbiome diversity, but an effect on immunoregulation. Is that correct? But then, given that we also know there is quite a strong link between this richness of the gut microbiome and the impact of metabolism, but also immunoregulation, is that evidence that we can dissociate the two, or there is something else that we should interpret?
Yeah. Well, as mentioned, you have a kind of microbiome dependent part. So some of the effects of food, let's say, go via the microbiome, but there's also still quite a part that is microbiome independent. So maybe all kind of metabolites are already absorbed in the small intestine, and then the interaction with the microbiome is not that evident. So I think both are there. There's a lot of focus nowadays on this microbiome dependent effects, but you also have to take into account that there are also microbiome independent effects that might be really relevant in telling the complete story. Yeah.
So pills is the next.
Yeah.
Nice. Okay, thank you very much. Thank you very much.